Yes, here we are again, I guess. Yes, everything seems to be working. Just a second. Can I hear myself? Yes, I can also hear myself. Hi, hello everyone, and welcome again to a microscopy live stream. Microbehunter here. Saturday always comes very quickly. Let me quickly at the beginning always check a little bit if everything is running properly. Yes, and I think we're ready to go again. And I would like to say hello all around the world. And I see that some of you have already joined in here. And I'm going to switch this from top chat to live chat to make sure that the sequence of chat messages is the same. Okay. So hello from Belgium, from Athens, Greece, Belgium, from Germany, Lithuania, London, Norway, Peru. Okay. England. So hello everyone. What you're able to see here is a live image from my microscope, right? You see a lot of paramecia, and if you're very lucky, look. I was really happy because some of them are actually also conjugating. So some of them seem to be double. I'm just look at this guy here. Do you see this here with the arrow? Yeah. Do you see actually they're joined together? Yeah. They're conjugating. This means that they're exchanging DNA. Yeah, now it's behind my head. And what I would like to do today is I would like to show you a method, a fairly simple method, where you can actually slow them down. And I tried this already today. I usually always do a little bit of an experiment during the day to prepare a little bit for the live stream. And I'm very happy to report that it actually worked out. I would like to show this method to you and how you can slow down the paramecia in a way that makes them much easier to be observed under the microscope. Yeah, so that's basically something that I will be doing today. So I'm going to take it quite easy today. I'm really happy. Look, there are so many conjugating. Look, they're here double, right? They're conjugating, yeah, and there's several of those. So that means they seem to be doing quite fine, yeah. So over here again, conjugating paramecia. I'll talk a little bit more about that later on. The point is the following. That especially the higher magnification, just want to show this to you here. They can sometimes be a little bit difficult to chase around and to observe because they're moving so quickly, right? So the question is how can you slow them down? And one way is well, you just use very little water and the cover glass on top, and this kind of presses them flat and squeezes them between the cover glass and the microscope slide. And then they basically cannot move because they're kind of compressed between those two glass surfaces, right? This does work. However, this kind of also deforms them quite a bit. And there is a possibility to now add a so-called methyl cellulose, which I just ordered. This methyl cellulose is very, very viscous. And when you add it to the paramecia, then, because it's so thick and viscous like honey, they cannot move quite as quickly, right? And then you can observe them better. And the good thing about that is, about the methyl cellulose is that it does not change, it's not osmotically active, it does not change, it doesn't kill the cells, right? So in other words, right? And that is really the advantage. And what I would like to, so that's as a little introduction. I see that some of you have joined in now. A short organizational message, again, is please, if you have any questions that are on topic and off topic, please post them. After approximately 1 hour, I will try to answer again the off topic questions. So please feel free to ask as many as possible. And as always, please also try to add us always as @Microbehunter or @Oliver so that I know that the message is directed towards me. Okay. And what we have here already from Hadrian, that is an interesting point that I was also thinking of. I've also tried different types of wallpaper paste. And indeed, wallpaper paste also contains methyl cellulose, but as you correctly say, they're not pure. And there's sometimes fungicides in there. And yeah, so that's the question of whether this is actually also working well. And again, here, you again are able to see several of those paramecia. Just watch them. Some of them are actually double. Yeah, so what they have done is they're conjugating. This means is they're fusing together to exchange their DNA. So that's a form of increasing diversity. Yeah, so this is actually quite something that I was quite happy to have found in this specimen. Look, for example, those two here. Yeah, they're joining together. And if you want to observe them better, we need to slow them down, obviously. Yeah? Oliver, can you show some of the previous preparations you've made before? I mean, using PVA and other permanent slides. I'd like to see how it preserved and looks like new. Maybe okay, yes. Bradle, thank you for the recommendation. Now, I can show them to you, but maybe not today, because I all locked them up in my cupboard. And for this reason, they're not accessible now, and then I'd have to stand up and collect them. But I'll basically remember this for an upcoming live stream, because over the last couple of weeks and months, I made several slide preparations. And basically the question is now, how are they doing? And they're somewhere still drying over there in my cupboard. Yeah. So yes, so I'm going to quickly show you what I've been doing, and and I'm going to switch now away from the microscope over to my desk view. And I'm going to just show you the finished preparation of today, but I'm going to make a fresh preparation so that you see actually how this works. This is a methyl cellulose solution that I made, and it's almost like a gel. Yeah, so I made this today in the morning at my time. So it's a 2% methyl cellulose solution, and I did have to wait a little bit for it to completely dissolve, because there's a real issue of them making clumps. And the idea is to kind of mix this with the paramecia, and then essentially they start to move more slowly because it's very thick. Right? Now the methyl cellulose, I also want to show that to you. As I already mentioned, or it was also mentioned in the chat, is a very common, also used in wallpaper paste. But again, there it's not entirely pure. It could be not pure. So what I got, I mean, again, this is not sponsored. I just bought this myself, and this was simply the one that popped up on Amazon first. Yeah, this is how it looks like. It says your methyl cellulose. Yeah. So again, I'm not. I've just bought this one, right? So not sponsored or anything, and it's actually used also as I think, if I'm correctly informed, as a food ingredient to thicken certain foods, right? To make it more viscous, right? So that's basically, it's 100 grams. This is really, I mean, it lasts an eternity. I think there might be the danger of it clumping because of moisture in the air. So one always has to keep it closed, and it says that you have to use it within a couple of months after opening it. I suppose that this is also due to the moisture, right? But in any case, that's the powder I'm using, and I did a little bit of research, and I found out that what you have, a good starting point would be a so-called 2% methyl cellulose solution. This basically means 2 grams of this mixed with 100 grams of water. That's a lot, right? So what I'm going to be doing is 0.04 grams mixed with 2 milliliters of water, right? So that's why I've got this here, right? And I'm going to, Wout is asking, could glycerol work as well? I think that it's even denser than methyl cellulose. Glycerol has a huge problem. And if you want to, I can actually try it out. I've got the glycerol over there. Glycerol really dehydrates the cells. So basically what's going to happen is that even though glycerol is viscous, it is highly osmotically active. This means the cells immediately start to lose water and they die. Yeah, glycerol, however, is very good if you want to see the individual surface of the cells, where you can actually see the cilia. So it dehydrates them. I actually want to make a separate video of this in response to last week. But glycerol, unfortunately, I found not to work as well, because you're actually killing the cells. right? Because they lose water, but methyl cellulose does not do that. Yeah, but glycerol has other advantages, especially if you want to do certain preparations. Yeah? So I'm just skimming the questions here, and you know what I'm going to do? I'm just going to start to show you the preparation, and it might take, I don't know, some time for it to dissolve. We're not going, if we're not that patient, we're just going to use this one, the one that I already prepared. Yeah? So yeah, we're going to simply start, and I'm going to do the following. I'm going to weigh first the methyl cellulose, and I'm going to weigh 0.04 grams. That's not a lot. Yeah? I always use those pieces of paper. You see, and then we could Tera. It's not very dense, so the actual amount is yeah. And dissolving it, oh, that's how it looks like. Look, this is just the powder, right? It's the methyl cellulose. I'm just going to use this here, yeah. And it's really, yeah, it's already too much. Okay, who cares? Yeah, 0.04 is would be the correct amount. Then we're just going to use a little bit more than 2 milliliters of water. It doesn't really matter, because as we use the methyl cellulose, and when we dilute it with the paramecia, it's going to change the dilution anyway. So I'm going to put this, yeah. And what you have to do is you should first add the water, and then sprinkle the methyl cellulose into it. Otherwise, it's the danger of clumps. Okay, and 2 milliliters of water. The problem with the methyl cellulose is that, of course, it doesn't store well, right? Once you've prepared it, you have to use it, because otherwise it might get moldy, yeah, and so on. Yeah. So somehow I have to now do the following. I somehow have to sprinkle it in here, and then at the same time I should also stir. Okay, you know what? I'm going to put the scale away, because I don't need that anymore. How do I turn this off? So now it's off. Okay, and I don't want to spill any liquid over it. Yeah, and I'm just going to sprinkle this in here, and yeah, and hope that it dissolves without forming too many clumps. Yeah, and you can already feel that it starts to slowly become thicker. It starts to absorb the water. Yeah. And then you have to be patient. Yeah. And again, we don't have to wait that long, because I already prepared this, and yeah, it will start to absorb the thing, and it will start to become thicker and thicker and thicker, and maybe something that might help. And I found actually that the tip actually is too narrow, because it's so viscous that it's not able to absorb. To suck up the methyl cellulose. So what I'm doing is I'm just going to cut this off. Okay, so it's easier for me to actually, yeah. Oh, can you see that this is actually? Oh, here we go. Okay, I'm trying to mix it like this. Yeah, and yeah, this requires a lot of patience, as you can see. And see, it's already starting to clump. But after a couple of minutes, as it continues to absorb it more and more, it's going to work. Yeah? But I guess, yeah, see, all of it looks already very sticky, because yeah, it's glue, kind of, yeah. Yeah. So, and then you have to wait a little bit for the bubbles to disappear. Yeah. Yeah. Not very, yeah. Not very nice looking, yeah. So you have to give it some time. Also, if you prepare wallpaper paste, you have to dissolve it for maybe, I don't know, some time, and simply wait for the methyl cellulose to absorb all of the water. Okay, you get the idea. Yeah, I mean, we can just leave it here right now to continue to let it absorb. And here we have a ready-made solution. As you see, it's pretty much free of bubbles, because they're all raised to the top, and it looks quite homogeneous. Yeah, so it's a question of patience. Maybe also warming it, maybe in a water bath might speed it up. But yeah, and you see already that the bubbles are starting to pop as well. Yeah? So I'm going to just give it a little bit of time again and see if there's some on-topic questions. Okay, so that was the question about glycerol, which unfortunately does not work, because it dehydrates the cells too much. Yeah. Would glycerol be better for higher magnifications and study of dead ciliates? It depends on the refractive index. Generally, glycerol as a mounting medium can be used and is used and has been used because of the refractive index. But I think what is really important is as soon as you mix it with cells, because of the osmosis, the cells are going to shrink. Yeah, so you're kind of losing a little bit the how do you say the natural structure. Yeah, so yeah, so I'm just reading here a couple of the you get the idea. So we're gonna have a look now on how the paramecia are doing again. So I'm switching back to the scope view. Here we go. It's a little too bright. Oh, look at this. The water is already starting to slowly evaporate, and let's just remember a little bit of how fast they're moving. Okay, and what I've been doing today in the morning is I simply decided to add pretty much the same amount of methyl cellulose to the water, and then you have to mix it, and this kind of worked, at least when I tried it, and then this kind of slowed down all of the cells, and we're just going to try that, okay, and look. I'm just checking a little bit the thing that I just prepared, and we're going to see if this works. And at least it did work a few hours ago. Let me check here. Let's take out the cells here. Here it is. Here, that's a little sample. In case you were wondering where I have it from, I put some pond water in here. And I added a little bit of a dry yeast and also some chlorella algae. Where did I get them from? These are tablets that you can buy. Actually got one over here. And these are some I don't know, food additives that they sell. And here you see these are green tablets of chlorella algae. And they always used to scratch one off, a little bit off, and use this as a food for the ciliates. Yeah, and it actually worked. You might remember that I actually have also worked with so-called wheat grains, which also are a very standard food source. So and now, big question. So it's around 25 mils that I have here. I might want to add a little bit more water here because I just realized it's not because it started to already evaporate. So I'm going to add a little bit more water. It's the same from the same sample. I'm going to now throw away the tip again because I want to use a fresh tip when I go into the methyl cellulose. And now the question is a little bit am I able to pick up some of the methyl cellulose, or has it already become too thick? So we're going to see that if it works. So I'm going in here directly into the methyl cellulose. And let's see if I'm able to withdraw. And it actually did work. Okay, you can actually see maybe carefully that there is, So this is, or is it? Yeah. Let me check. No, actually not. Okay. It's too viscous. Okay. No, it didn't. It didn't take anything. It was too viscous. So what I'm going to do is the following. I'm going to again take a tip, and I'm going to cut the tip off, simply to make it easier for the liquid to be taken up. It's actually not a liquid anymore. It's almost like a gel. And now it works. Okay. Yeah. Now actually it works. There is it's in here. I close this, and I'm simply going to add it now to the paramecia. Look, there is a light lamp burning here. I'm going to try switching it off. So that's better, otherwise there's a reflection. So I'm just going to add it now to this, and I'm going to carefully pull it in, and out, carefully mixing it. Okay, you see there are indeed some bubbles. Okay. And maybe this is enough. I hope it's enough. Yeah, those bubbles here are a little bit annoying. However, they will disappear. Hopefully, as the methyl cellulose in the liquid kind of settles. Alternatively, I might just drop a cover glass on top and then hope that the bubbles are pushed to the side. Let's try that. It's pretty thick. Yeah, it's pretty thick. Let's see if this actually works. Yeah, some see the bubbles are popping. That's actually good. And I'm going to put it now directly under the microscope, and I'm really going to hope for the best that we're able to see it better now. I don't know. We're going to see. And here we are. Okay, here's already one of them. Yeah, it's being pulled along. Okay. I have to here. Yeah, here this one is these are conjugating. It might have been a little too much. Let's see if it's still alive. Yeah, you can still see the cilia beating. So this one here is conjugating, okay? So they're and I'm going to make it a little bit open up. You see, as the bubbles burst, they basically are being pulled along. I think I've taken actually more of the methyl cellulose now than before. Yeah, but we can now see much better also the cilia beating. Okay. And you can also see that the shape is retained. Again, these are two paramecia. I need to explain this a little bit if you don't know the biology behind that. What they're doing is they're sexually reproducing. Yeah. So paramecia, of course, they reproduce by cell division. But when they reproduce by cell division and when they divide, you're not really increasing genetic variability. Yeah. But the point of sexual reproduction is to increase genetic variability. So what they're doing is the two cells are fusing together and they're exchanging the DNA with each other. Yeah, they have a so-called a macronucleus, which is the main nucleus, and they've got so-called micronuclei, which basically contains DNA that can be exchanged directly. So they're actually fusing together, so the cytoplasm is also fused, and then they can exchange it. And I was kind of happy that to be able to see it like this, because otherwise they would be swimming around so quickly that you're not able to see them. Yeah. So so that's basically, and you see also over here the other ciliates over here, they're swimming around much more slowly now. Yeah. So that's quite nice. Let me go down with the magnification very low. So let me see this so that we're able to get a better overview of where they are. So that's the one over here. It looks almost like it's not moving at all. Yeah. There are a few more over here. Oh, they're really crawling along slowly. Yeah. Let's zoom in here a little more. Yep. And here they are. So they are really struggling to move forward now. It's almost like time lapse, a slow motion rather, right? Yeah. And really, the good thing is that the methyl cellulose does not kill them, because that's And because the cilia are also beating much more slowly, you can actually also see them much better. So here we go. So, do I understand correctly? Paul is asking that the micronuclei contain the same DNA as the micronuclei. No, it's that's the reproductive cycle of the paramecium is really complex. Ultimately, the macronucleus will form out of the fusion of the micronuclei. So what happens is that the macronucleus will form micronuclei, and the micronuclei will undergo meiosis. This basically means that they're different micronuclei. The genetic information of the micronuclei is different than the macronucleus, and so there are two micronuclei, and one of them each is exchanged with the other paramecium, and then the micronuclei after the exchange they fuse together with the existing micronuclei. I actually have to make a drawing of this. So and then when they fuse, you get a diploid nucleus which will divide several times. Several of them will degrade and degenerate, and then out of those fused micronuclei you have again a new macronucleus. And then this one will again form micronuclei. This was such a long time ago. I had to study this at university long time ago, and I just recently again looked at the at the reproductive cycle of paramecium. It's pretty complex, really. Yeah, it's really complex. But the idea is those cells, they need some kind of a mechanism to increase genetic diversity, because otherwise, if they're just dividing, then the offspring are genetically the same, right? And that's not good, because you need to have a certain genetic diversity within a species. Otherwise, the species is not able to survive a changing environment. So they have this mechanism of actually exchanging those micronuclei with each other, out of which a new macronucleus is made. Yeah. And the thing is the following, that normally what you have to do is you have to use, they discovered this apparently by using staining techniques. So they were staining the DNA, and that's how they discovered the macronucleus and the micronucleus and all these things. Yeah. So do the micronuclei act like sperm cells? Yes, as a matter of fact, that's correct. Yeah. It is correct. The micronuclei, because they are formed by meiosis, essentially they are similar to sperm and egg cells, like gametes, but they are actually directly exchanged from cell to cell. And so in that sense, there is indeed in eukaryotes all have this. They have the same principle of having meiosis happening, where essentially you've got a haploid cells being made, which then again fuse with another haploid cells. In paramecium, it's not like this, that you have male and female, but you have so-called mating types. Yeah, I don't know how many there are, but they have to be of opposite mating types for them to actually conjugate. Yeah? Yeah, so hi, I finally caught you live. Is this? It's DIC, but it's not a BH2 microscope. It's a BH. It's a BH 53 microscope. Yeah? It's a slightly more modern version. Yeah. So you see also that the other cells, the other protozoans that are floating around are much slower right now. I mean, what we could do is we could actually try to do this again with a different amount of methyl cellulose. Maybe, yeah, but it's okay, I guess. Yeah, because you see that it's slow enough that we're able to follow it properly. But let's go again down to a lower magnification simply to get a better overview. Yeah. It's so much easier to observe. It's not even a comparison. Yeah? It's not even close. What is this here? Yeah, it's another one over here. Here's another one, almost crawling along like those snails yeah. But I think they're also easier to overlook, maybe, because when they're whizzing around on the microscope slide, they're easy to see. But now they're a little bit more difficult to, because they almost look stationary sometimes. Yeah. So every now and then I'm checking the questions. I know that paramecium are supposed to have two pulsating vacuoles. I always seem to have a hard time finding them. Could you try to point them in here? Yeah, you're referring to the so-called the contractile vacuole. I actually did make a few, some time ago, some videos about this. The contractile vacuoles are actually better seen when you use very little water, but I'm going to try to do that without methyl cellulose. I'm just going to show you now how you can slow them down using very little water as well. And as a matter of fact, I do have also over here some nematode worms, so-called vinegar eels, that really move very vigorously. And I would like to also put them under methyl cellulose. But let's do the following now. Let's have a look at those contractile vacuoles. Okay, just because you're asking, and why not? And this way I can also show you, I can actually also show you a little bit a second way of slowing them down. Let me see how the desk here. Yeah, so yeah, I'm going to take this here again. This sample is quite okay. I need a fresh pipette tip. And I actually found out that over here, most of the paramecium are not found on the surface, but some, for whatever reason, the bottom. Not for whatever reason, but because over there you have the food. Let's see if we're, you know what, I'm going to just use a fresh. I'm going to use a fresh slide. I'm just going to use a fresh slide. Okay. when you're trying to compress them between the cover glass and the slide, the amount of water is really important. If you use too little water, you're actually going to crush them and the cells are going to pop. Okay, so let's see if there are any cells here first. I always do a very quick check first without a cover glass, simply to check if paramecia are present enough, and I think sure they are. Okay, plenty of them. Okay, so that's good. And now I'm going to directly drop a cover glass on top of them. Basically, yeah, trying to compress them. And here, this is a little dangerous now, because if there are too few, if there's too little water, then there is really the danger that I'm going to crush them. So what a technique could be that you add a little bit more at the beginning, okay, and then remove a little bit using some tissue paper. That is essentially also a strategy. And when they're really pressed flat, then you're able to see the two contractile vacuoles much better. Okay, so let's just drop a... This is not a nice looking cover glass. It's a little bit dirty. Let me quickly... It's a little bit of pity because some cover glasses are really, I don't know, like they're covered with this kind of a grime or grease, or I don't know. Yeah? So here we go. This is quite a plenty of water, because as when you see that there's water coming out from the side on the edges, is actually a sign that it's probably too much water, but that's okay. We need to, where is the scope here again? Where is it? Here we go. Okay, yeah, you see that with the cover glass is still floating around, whizzing around quite a bit. This is a sign that still too much water. Okay, so they're able to move quite freely. And you see that the cells are quite elongated. So what I'm going to do now is I'm going to take a tissue paper. You know what? I'm going to not do this under the microscope. I'm going to do it on the desk to show you what I'm doing now. And we're just going to remove some excess water from the edge here. Okay. This is even. Am I even touching this? Yes. Here now we go. Okay, a little more. No, this doesn't work. I'm not touching it properly. Here we go. Okay. And maybe here on the side as well. And okay, let's give this one here a try. Scope view. Those takes, yes, see here, that's actually quite nice. You see now that they are much more oval, the cells. Okay, let me go higher with the magnification, and they are now a little bit compressed, but not to the extent that they are already, yeah, the cells are popping open. Okay, so good. Let's move up with the magnification, and maybe we're able to see. As a matter of fact, I already see them, the contractile vacuoles that you were asking for. So, and I already see, okay, did you just see this? Okay. Up, here we go. Where is the arrow? Yep. Have a look at this here. This is one of the two contractile vacuoles, and what they do is they pump out water. Water moves into the cell by osmosis. Okay, and then you see that how it contracts. Yeah, it fills up again. Yeah, and maybe there's another one visible here in front as well. But this one over here. Yeah, and do you see those radial canals going out? That's also very typical. Yeah. So, yeah. So, yeah, see, this contractile vacuole over here is quite nicely visible, yeah, because you just wanted to know how it looks like. Yeah, here it is. So I'm using now 40 times, objective. You can actually see it quite nicely, also in phase contrast. So I'm also going to show that to you now. So that's, by the way, that's the second way of limiting the movement. And how? Oh, I lost it. Let me quickly check over here. Where is it? Where is this? Here it is. Here it is again. Okay. Yep. And here you look at this here. That's one of those and over here the other one. And those radiating lines are the radial canals that are also needed to collect the water, and then they're pumping out the water. Yeah? So two methods: use methyl cellulose or yeah, a little bit of water so that their movement is limited. This is the oral groove, what you see over here. If you actually look very carefully, it's going to suck in food, and then sometimes you're actually able to see how a new vacuole starts, food vacuole is starting to form. Yeah, so this is quite, some of these bubbles that you see here are food vacuoles, and they basically contain food which is being digested. And what you're able to see sometimes, the food is being sucked in, and you see a little new food vacuole starting to form here, which then is kind of pinched off, and then it's in the cell. Yeah? So that's in phase contrast. If you don't have phase contrast, many of you might not have phase contrast. That's okay. This is how it will look in bright field. And you see that you're still able to see quite a lot. Okay. That's yeah. So all of the structures are still obviously visible, and yeah. Let's do the following just for the fun of it. Let's go all the way up to 60 times. And I have to really go up with the brightness, otherwise it's too, so I've got 60 times magnification. I think it's really, I don't know, it's really cool of what we're able to see with modern microscopes. Yeah, all of this, the cellular events and so on. Yeah, and if you basically want to, make them move again a little bit more, look, what I'm going to do now is I'm just going to add, a little bit more water. Because if I wait too long, then they're going to die because the pressure is so high. So I'm adding now some water. Interesting, where is the stage? I thought that I have a stage as well. Just a second, let me quickly check. I'm a little bit surprised because I used to have the stage as well. Here we go. Okay. Let's make it maybe a little bit smaller so it doesn't disturb. Okay, I totally forgot about the stage here, so that you can actually see what I'm doing. Let's remove the arrow and have a look. I'm adding now here on the side, I'm adding a little bit of water, of course, right next to the cover glass. And this was too quick. Yeah. And immediately because the film of water increases in thickness, then they start to move away immediately again. Yeah, because they have more freedom of movement. Yeah, I mean, you get the idea. Huh? So let's have a look here. I'm just going to respond to the questions that might be topic related. All of the other questions or comments I'm going to talk about later. Okay. Is the product usable for bigger species? Bigger species, like water fleas, maybe? Why not try it out? Okay. So that's kind of interesting, yeah. And there are some questions about filming them at normal speed and then magnify and slow the speed. I'll respond to that. Generally, I've tried this as well. That essentially to slow down the video, but it was blurry. That's the problem. Yeah. So in principle, yes, but because of the shutter speed limitation, therefore it was actually, when you slow it down, it was kind of blurry. Yeah. Yeah. I mean, you get the idea that as soon as you add a little bit more water, then they start moving again. Yeah? What are we going to do now? I think I'm just going to try. Yeah. I'm going to try to put some. Yeah. I'm going to try to add again a little bit of this methyl cellulose solution in here. What time is it? 38. You know what I'm going to do? I'm just going to add a little bit less maybe because I would like to change the... Let me put this here. Okay. Let me put this away here. And let's try another one. Maybe by changing the amount. Yeah. Let me try it again. So this is now 25 mils. Ooh, that's a lot of stuff here. Okay, and I'm going to change this. Let's go down here. I don't know. Let's go down to 10. Okay, 10 mils. In case you're wondering, this is how it looks now, the one that we made before. You see it's much more clear, but still not fully dissolved yet. Yeah, it's very thick. Yeah, yeah, so I think maybe warming it up might also help to speed it up. Yeah, or you just wait an hour or so. Yeah. But it's okay if the clumps are not too bad, then I think it's fine. So, and I'm going to cut off the tip again, and I'm just going to use some of this one that I prepared already before. Now let's try to use a little bit less, and let's see if this okay, has another impact here. So it's actually okay. Yeah, you can already see that it's a little bit thicker. And cover glass, let's drop more cover glass again on top. And let's see if the movement speed is a little bit different here. Scope view again. It's too bright. I don't see any. Where are they? Here's one. So let me quickly check. Yeah, see there, they are moving a little bit faster because it used a lower concentration. Here we go. Yeah, so essentially you can play around quite a bit. Yeah. It's kind of interesting where I did not seem to have a lot of, maybe I just happened to grab a sample or a corner or a batch where there were fewer. Okay, here are more. Yeah. Okay, I think we've watched enough paramecia. Now I would like to move on, and I would like to try now some of the nematode worms. Yeah, that I kept. Yeah. That's really, I have to say that this solution is really convenient, I think, because it's so easy to use, and the cells really seem to be very unaffected by it, other than the speed of movement, right? Yeah. It's almost crawling along like worms. Yeah? So, just for the fun of it, let's move on, because I'd like to try something else. And the next thing I did not try yet. Okay, so this is going to be interesting. I've not tried it yet, and I did not put these vinegar eels under the microscope now for quite some time. Those of you who've been watching some of my videos know that here this is basically, these are vinegar eels. I got them a couple of years ago from a colleague who was kept in an aquarium, and he basically is growing them as fish food. Yeah, vinegar eels are simply nematode worms, and I think they belong to tubifex or whatever. And they often like to be on the surface. Let me go up and let's go to 20 microliters, and I hope that they're still alive. Okay, but there should be, and I'm going to just grab a small sample from the top on here on the microscope slide. No cover glass. Let's just see if there are a few of them visible. This smells pretty bad, like vinegar. Where are they? Okay, here they are. We've got still a few of them. Let me flip this out here. Yeah, so these are basically, also very difficult. I mean, fun to observe, obviously, right? But let's also try to slow them down. And I do not know how they react with the methyl cellulose. Okay. So I also hope that their movement can be slowed down a little bit. I'm almost quite sure. But yeah, they like to be around in a certain pH. So I don't know if by adding the methyl cellulose, how the acid of the vinegar, how it interacts with the methyl cellulose, or yeah, if it changes the pH around a lot, I don't know. Okay, so this is something that I would like to try. Okay, so let's add a little bit of this solution here as well. And let's see if this kind of slows them down. So where is the? Never forget to switch back to the. Yeah. So this was not 20 mils, microliters. I don't know. Let's. Yeah, it's so viscous that we always catch more anyway. Let's try 10. And again, this is here the first try. Okay, so this is not highly experimental. I've never tried this here. Let's add a little bit of this. And I don't know how strong they are. If maybe they have so much, so strong that it doesn't really matter whether we add this or not. But we're gonna see. So, carbo-glass, always make sure that you only get one of them here. Oh, this looks so thick. No, it is one, but I'm going to wipe it again. And I don't know, it almost looks like it's 2. Yes, it's 2, of course it's 2. Here we go, let's just drop one of them on top here. Hope that the air bubbles are being pressed out, and let's see, okay? Scope view again, and aha, here it is. Here are 2 of them. 3, and let's zoom in. I mean, their movement is a little bit different. They used to move back and forth, and now they're kind of curling up a little bit. Yeah, but I think it also works. Okay. Yeah. So we can see that this methyl cellulose is evidently, yeah, also useful for slightly larger specimens, as you were asking. Yeah. Actually, kind of creepy if you just look at it. Yeah. Ah, somebody is greeting me from Altmünster. Haha, hello and welcome. Yeah. Joining the live stream. And what I've done here, for those of you just joined in, these are nematode worms, which basically I got from a colleague. I'm a teacher in school, right? And there are many people who have fascinating hobbies in my school, and one of my colleagues is also keeping an aquarium. And some time ago he gave me those worms here, and what I've done now is I've slowed them down by adding so-called methyl cellulose to those worms, and those worms now start to move slower, and now it's easier for me to observe them. Yeah, they look scary. Yeah, but they're not scary. They're completely harmless. But having said that, there are, of course, certain nematode worms out there that are parasites, and some of them indeed can cause certain diseases. Yeah, so trichinella, for example, in uncooked pork, it can be a problem. And there are a lot of these worms out there, right? And as a matter of fact, I think I forgot his name. There was this one worm scientist back in the day, 19th century, I think, who said is if you remove everything from planet Earth except the worms, you're still able to see the continents and everything, yeah, because there are so many of them. Yeah. So in any case, I'm going to go down a little bit with the magnification again so that we can see more where the others are. You see, because I added the methyl cellulose solution, therefore, of course, I diluted them a little bit, and they were kind of pushed apart. Here's some more. Yeah, I sometimes feel that a little bit. It's almost creepy, like like in a nightmare. Yeah. So let me increase the brightness a little bit. Yeah. So that's actually to a certain extent they do look kind of interesting and beautiful and beautiful. I don't know. Beauty is in the eye of the beholder. Yeah. Oliver wondering if you can use half water and vinegar with methyl cellulose. Half water and vinegar. So basically, what do you mean half water and vinegar? So do you mean that I prepare a methyl cellulose solution already with vinegar? Is this what you want to say? I'm not fully aware of what you mean. Half water and vinegar. Or do you mean that I dilute it yet further, maybe? Huh? Yeah, so will they make you sick? Well, those won't, those specific ones here. Definitely, I mean, if you swallow them, they're going to be just digested, right? But there are certain, it depends on the nematode that you have, but there are certain worms out there which essentially which are known as parasites, okay? And which, Trichinella, for example, but yeah, but they won't make you sick. Huh? They actually, if you keep vinegar, it can be quite possible that it can become contaminated and then they will start to grow in it. So that's nothing unusual. So if I were to eat those, yeah, they're just going to be digested. Yeah. So, yeah. You should be careful when you eat sashimi. That is indeed the case. How do you kill those? So if you eat raw fish, for example, in Japanese food, what they will generally do is they will freeze. They will freeze the fish, and this will actually kill any nematodes, so there are no issues. Yeah, and there are so many of them around. It's almost impossible to avoid them. Yeah, I thought I could use vinegar to clean with. You can use vinegar, of course, because vinegar or strong low pH will essentially kill indeed many organisms. But essentially, yeah, it depends very much on the organism. Yeah, and you cannot avoid them anyway. Yeah, but they like vinegar. Yeah. Yeah. So let me simply slow down again. So this is a little bit the thing that I just wanted to show you, that this methyl cellulose is really a nice little trick to make things visible. But because I added a cover glass and because I added more of the solution here, they're spread out now much more. Yeah. You know what? Just for the fun of it, I'm just gonna show you. I'm gonna try something else. Because maybe I'm able to grab a higher concentration of those little critters from the edge here. I'm also going to cut the tip again. Oh, I dropped the scissors. Okay, here we go. So, let me see. Okay, just a second. Let me see. You see, all these things here on the side might be, I don't know. Let me try to grab some more here. Maybe we're lucky. I don't know. Let me quickly check. No, not so many. Not happy. Let me see if I can find some over here. All of this white stuff that we have here. I don't know. I'm gonna give this here a try. Scope view. Yeah, there are a few more here. Okay, it's actually. Yep. Here are a few more. Okay. And yeah, let's simply repeat it a little bit with a few more of these nematodes, and let's see if we're able to slow them down again. Let me simply put the lid on top here, put this away. I don't want to tip over the jar. Okay, and let's add again a small amount of this. You see, the point is the following. I'm a biologist, yeah, so I don't feel so extremely disgusted. I remember just a short story. I already told you I'm a teacher, and students, especially young students, they sometimes have a surprisingly, how do you say, relaxed attitude to certain things, right? And I remember a few years ago, one of my students, they have had pets at home, and they basically pulled out the ticks from the pets, and these were large, soaked full with blood, and they looked like berries, right? And the student essentially dropped it off on my table in a glass in a glass jar like this, and it looked like berries, and a whole, I don't know, 10, 15, 20 of those, simply because the kids were fascinated, and they had their little pet animal, dog or cat, and pulled them out and just gave them to me because I'm a biology teacher, and just had them standing around on my table, and not how do you say, minding very much about it. But the teachers around me, they didn't like that. They looked at it and said, "What is this? Why are you going to eat those berries?" And then they realized that actually these are ticks because of the legs here, and they were really upset about that. Yeah, so it's a little bit of a, how do you say, yeah. For me, it was completely natural. Who cares? Just a whole bunch of ticks, totally normal. But not everyone likes these things, and that's a little bit maybe in that sense I'm maybe a little bit of a nerd, Yeah. So I don't know if this is enough really, but yeah. So let's mix this here a little bit. So I used now slightly less of this methyl cellulose in comparison to the water. Let's give it another try. Let's drop it on here, and let's have a look. Again, under the microscope. Yeah. Here we go. Yeah. They are much slower. Okay. Just seeing. I just hear that essentially. Thank you. I got the questions. I got your questions again. Here, yeah, which I'll be answering in a few minutes. Yeah. So that is essentially it shows that it seems to be working. And for those of you who joined in a little bit later, in today's live stream, I showed you a way of slowing down cells, paramecia, and also those worms here by adding a so-called methyl cellulose, which is very viscous, like honey, and because it's so thick, the animals and the cells are not able to move quite as quickly. Yeah, and this makes them easier to observe under the microscope. Yeah. Yeah, a comment here. That is just so great about your hobby that show us that some creepy things are quite fascinating. Yeah, it's a little bit. I'll tell you another thing. Every year we're during the biology lesson we're doing a so-called an insect project, and the task is that students we are organizing an entomologist. He's an insect specialist. He's been cooperating with our school now for many years, and he is catching insects with us in the school garden, and we put them under the microscope, and he helps us identify them. Yeah, and because some insects are protected, he basically helps us sorting them out and giving us advice, because it can become a little bit complicated. And the reason why we're doing that is because it's an insect, and how disgusting. But actually, if you look at them under the microscope, you're getting reconnected to nature a little bit again, right? Because we're exposed to insects all the time, right? But we just don't look at them in detail. But when you put them under the microscope, you're actually realizing how complex they are, and yes, even how beautiful they can be. And we've received the feedback from some of the students that ever since they've done this insect project, they felt less afraid of insects. Yeah, because some of them, they were kind of disgusted, and there's a little bug somewhere, right? And they didn't like that, and it's considered dirty or whatever. But once you're a little bit exposed to the whole thing, you have a more natural and relaxed attitude to those. Yeah, so that is a little bit, And the opposite, after the face mite video, I can't sleep anymore. Yeah, same thing. That's with the face mites, which basically pretty much all of us have, right? But it's psychological. Simply the idea that there are little animals living inside the hair follicles of our skin, yeah, is not a comfortable idea for many. And I say, who cares? Because it's just part of who we are. As a matter of fact, we have more bacteria growing on our body and in our body, bacterial cells, than we have body cells. So actually, we're made more of things that actually is not us, if you were to see it like that. Especially in our digestive system, our digestive system is a massive bioreactor, right? And contains more bacteria than we have body cells. So and that is and if you are into biology a little bit, and you're just getting used to these things, it's So and as we look at those more or less, look, they're almost making a knot here. Yeah, and I'm just going to go through some of those questions here. Okay, let me quickly, if you want, so I can quickly show them to you, or we can continue to watch the video here. Here are your questions. First one is, I'm using methyl cellulose from the hardware store. Any difference from the medical form of the product? First of all, what I have is not medical grade or laboratory grade methyl cellulose. It's it doesn't have to be. It's I think a food additive for cooking, so to make food a little bit thicker. In that sense, it does not have to be pure. First of all, because the liquid or the sample that we have is not pure anyway. So any form of other substances we're adding anyway, indirectly, then because of the sample that we're adding. However, the one from the hardware store, we just have to be aware that if you're using the wallpaper paste, that sometimes there is indeed some fungicide included. So basically that prevents the wallpaper paste from becoming moldy. I don't know if this has an impact, and the answer is very, very simple. Try it out. Okay, that's generally a recommendation that I have to give. It's a hobby, and my philosophy is not to use everyday materials as much as we can. Yeah. So that's the thing. Do you happen to know any good books on mold fungus identification? Thank you so much for your work. I can only tell you that what I've heard. Well, first of all, if you want to identify fungi like the mushroom, the fruiting body, basically because you want to eat them for food, there are plenty of books available. But my suggestion is you got to be careful. Seriously, okay? So that is really a thing. Fungi can produce extremely potent toxins, and if you don't really know what you're doing, and sometimes you think that you identified and you think that this is a characteristic that you're looking at, but it's not. Okay, so you have to be. So I don't know specifically any good books. I know that there are books available, but if you now want to go into mold identification, so basically on the microscopic level, under the microscope, if this is what you're interested in, then I have to tell you, then things start to become really difficult, okay? Because identification of microorganisms using a microscope is very limited. And what I heard while I was still at university, it's a really tricky thing, because depending on the environment, the mold will grow differently. So apparently the environment has a very huge impact also on the way that the mold appears. Yeah? So for that reason, if you really want to identify it properly, you got to be a real expert on it or do DNA studies. Yeah, but simply because the biodiversity is so high and the microscopes provide so limited information, for microscopic mold, it's interesting to look at. Maybe you're able to go a little bit into a certain direction of what it could be. You can, of course, distinguish septate from aseptate fungi and so on. But anything much more detailed, you have to be really probably use specific staining techniques or other chemical tests. Yeah. So the micronuclei act like sperm cells and egg cells in higher animals. Yes, indeed. Yeah. So that's they are formed by meiosis, just like sperm and egg cells. and they will actually fuse together and to make again a diploid nucleus. Okay, so that's indeed correct. Yeah. And that's also a form of sexual reproduction, with the difference that in paramecia, as we just looked at, this exchange of DNA is separate from the actual reproduction of where the cells actually divides. Yeah, so the division and the exchange of DNA are two separate processes. Maybe far-fetched, is there a way to film that at normal speed and then magnify and show speed the video for big screen replay and recorded video? So the question is it possible to kind of slow them down? Okay, that's the question. I have tried to do that. What you need, and I tell you that's a little bit in principle possible, but you need to have a very high speed, sufficiently high speed camera, obviously, and yeah, a lot of light. Yeah, so my camera is currently recording at 30 frames per second, but if you really want to have catch the fast movement, you need very short shutter speeds and so on. I don't know, maybe it would make more sense in this case simply to, I know that some mobile phone cameras, some iPhones and so on are able to record at 120 frames per second, and it might actually make sense and to directly connect maybe the iPhone to an eyepiece or the photo tube and do it that way, right? But my camera system is quite a nice setup, but is only able to record right now at 30 frames per second. So I'd have to see if it actually, maybe if I lower the resolution, it can go maybe to 60 frames. Yeah? But generally it is possible, yeah, but you need a lot of light for that. Yeah? Is it normal for isopropanol to leave stains? Because I've recently cleaned all my glass pipettes with isopropanol alcohol and it left slight crusty stains. Shouldn't. Actually, the alcohol should completely evaporate. So what I can imagine, and I'm just hypothesizing, is that maybe the alcohol started to dissolve something, and that basically what the deposit that you're seeing is basically the dissolved substance that was the residue which was left over after the alcohol evaporated. But actually, unless it's pure isopropanol, it should not leave any stains, or they added something to it. Yeah? But more likely is that maybe it, you accidentally dissolved something. Maybe there was some mounting medium left, and kind of this kind of got spread into a very thin layer, and then this was actually left over. I would have to guess here. Yeah? Would it be nice to calculate how large these vacuoles are and the extension of how water is pumped out per minute? You can calculate that indeed with a micrometer slide. And I know also that people, I have not done this, but people have actually changed the salt concentration of the surrounding medium. And then they were actually also able to determine how often they pump. But actually how much it pumps, I don't know, but I guess you could estimate this by assuming that this contractile vacuole maybe is actually a sphere. Yeah, so it should be possible to actually indeed calculate that. Yeah. I wanted to thank you for introducing me to the fascinating microscopic world. This channel inspired me to buy my own microscope, and I'm now studying to hopefully become a biology teacher. Now that is something, really. No, okay, I mean, honestly, if you're interested, I could do a complete video just on about the ups and downs of being a biology teacher. Okay, no, I'm happy that it inspired you. Yeah. Just be aware that biology in school and at the university can become quite dry. Yeah? So make sure that you're keeping the lessons interesting. Yeah? And that's also one of the reasons why I'm making those videos, because I can show them actually also to my students. Yeah? These nematodes move crazy. Why? It seems to take a lot of precious energy. Yes, is that what you're asking here is with a why? What is the evolutionary advantage, right? And they're moving so quickly, and what's the point? Yeah, of them moving. Let me quickly see if I'm able to find a few of these guys again. Yeah, what's the point of them moving? That's not one of them. Yeah, and the short answer is by moving, they're agitating the surrounding water or medium or vinegar, and this way I can imagine that they are moving the food around. Yeah. for eating. So movement is a very important characteristic of life, and all organisms move, even plants move. If you go into look into the cell, you can actually see that there's quite a bit of movement inside the cell. So that is one of the typical characteristics of life, yeah, locomotion. And what they do actually is they will move towards oxygen. So that's why in the jar they were actually on the surface, yeah. But generally, yeah, every organism has its own strategy of survival, yeah. So, wondering if you can have water and vinegar with methyl cellulose, basically maybe dilute it down a little bit. It would be interesting to find out whether the contraction process in Vorticella can be better observed with the help of methyl cellulose. And this is indeed something that I've also was thinking about. Vorticella is actually one of the fastest movements known. Yeah? It's a ciliate on a stalk. And if disturbed, it really contracts so fast. I tried to film it in slow motion. It's impossible. It contracts so quickly. It's immediately gone. So this is indeed something. Maybe we're able to kind of slow this down as well. Oh, look where the worms have gathered. Maybe over there where there is more oxygen. See on the side of the cover glass. Yeah. So that's indeed something. I don't have any Vorticella right now. Otherwise, I would have tried it out. But indeed, that is one thing that I have also on my list. Yeah. So let me quickly check again over here if there are some more questions that kind of appeared. Yeah, okay. Particular thanks for the awesome phase contrast view at high magnification. We could see the vacuole canals in action. Yes. Very nice. Have you ever tried Hoffman modulation contrast? No, I have not tried it. I've read about this, but my microscope currently does not allow me to do that. I'd have to tinker around a little bit. Yeah. Okay. Can you show us E. coli? The short answer is yes, but not yet. As a matter of fact, we've grown E. coli in school. So one of my students is doing an experiment using E. coli. And where did we get them from? We got them from tablets. We ordered some of these capsules containing E. coli, and those capsules are swallowed to regenerate the digestive system after, for example, you've been on an antibiotic treatment. When you have swallowed antibiotics, then this will kill off a lot of bacteria. As a side effect, so to say, in your digestive system, and in order to regenerate that, you can buy those freeze-dried E. coli bacteria and you swallow them, and this kind of will regenerate the digestive system, so they're safe to use. Okay, and we've actually grown a few of those, and indeed this is one of the things that I also would like to show you, maybe in one of the upcoming streams here. I'm a little bit reluctant in working with bacteria, generally in growing bacteria, because I know that there are a whole bunch of folks out there on YouTube who actually are growing bacteria for fun and so on. And I've seen some videos and I said, that's not safe. That's not safe. Especially if you're trying, very common where you try to grow bacteria from your hand, you kind of press them against an agar dish to show the bacterial colonies from the hand. You shouldn't be doing this stuff, right? Because you're really growing bacteria at an unusually high concentration. I don't mind growing so much E. coli bacteria. You still have to be careful of contaminants, but growing bacteria that are actually growing on your skin and so on, Staphylococcus, I don't feel comfortable with that. And I'm a little bit worried that if I actually show you these things, even though I'm a trained microbiologist, I know how to observe the safety procedures and so on, because we've really been trained. I worked in a safety category 2 lab. There are 4 safety categories, and I was in a category 2 lab, so it's elevated security. Because we were working, I was identifying unknown bacteria, and you don't know what you're growing. Yeah, it doesn't mean that they're automatically dangerous, but you don't know what you're growing. Yeah, until you've identified them, right? And in many cases, you're discovering new species, right? You don't have to assume that they're automatically almost always dangerous, but yeah, if you don't know what you're growing, it's like eating mushrooms that you don't know, right? I mean, I'm not eating the bacteria, but I mean, yeah. accidents, lab accidents can happen, and bacteria can spread around. And so for this reason, I'm a little bit reluctant in kind of showing microbiological techniques, on my YouTube channel, just because I'm kind of worried that people are actually gonna, do this as well, and maybe a little bit improperly. And you do have to be a bit careful. You also have to dispose of them properly. And so on. Having said that, if you have got some kind of a container somewhere where you put the vegetable trash and kitchen trash, and you decompose it and compost it. I mean, this is also quite a container growing bacteria, right? Yeah. So yeah, I'm going off a little bit here on the side. After the face mite video, okay, we got that. Yeah. So, how could you specifically raise or grow Vorticella? It's kind of fun, interesting. Sometimes, let's put it this way: I've grown over here. I just show this to you what I've done, and then sometimes, if you're lucky, you're also growing Vorticella. Sometimes, if you're lucky. So what I've done here is the following. I've got a water sample, and I added a little bit of food. Again, you got to be careful because you're growing unknown organisms. But the paramecia that will grow rapidly will kind of keep the bacterial count fairly low. And occasionally what I've seen is I've also seen that towards the surface, sometimes colonies of Vorticella will actually form. Yeah? And this is something that is a little bit I guess a question of luck, because it depends on what original organisms were present in the first place and whether the conditions are right for them to reproduce. I've already done a couple of tries, and I was not able to enrich any protozoans. I was kind of surprised. What could it be? And then I discovered that there were a couple of water fleas in there who might have actually eaten those Vorticella. Okay. So just saying, talking about water fleas, I'm going to now do the following. Maybe this just as a last experiment. Just a second. Give me 10 seconds. Okay. Look what I've got here. I don't know if I have to check them. Yeah, you see, I don't have my reading glasses with me right now. I put them somewhere. See, I'm already. There are Daphnia in here, and where's the pipette? And they can be a little bit difficult to catch. Let me see if I can find some. I've been feeding them now for months here. I can't see them. That's the problem. They're so quick. I know that they're floating around here, but let me see if I'm a little bit lucky here. I don't know. I don't know if I actually got one here. Let me see. Nope. Or yes, I don't know. As a matter of fact, I might have got one. I don't know, not only Daphnia, but also some other. We could try to. Yeah, there might be one. Okay, let me try this here. Maybe we're lucky. But kind of slowing down the movement of water fleas is not such a big deal. You just use very little water, and then they're not able to escape anyway. Let me quickly check. Nope, unfortunately no. It was another piece of. Yeah, they're kind of difficult to catch. It was just some dust or dirt. Let me try this again. Because they immediately escape. Let me see again. Because this would also be kind of interesting to kind of check whether they are able to, if you can slow them down. No, I think I'm going to. Nope. The density of the. Is this one? Let me see. I gotta be a little bit patient now. Okay, I'm not talking a lot now because I'm concentrating. So let me try this. Yep, here is one. Here we go. Right? Okay, got lucky here. And okay, I'm a little bit relieved that this actually worked out. I was able to catch one of these guys. And now the question is are we able to slow them down as well using some of this methyl cellulose solution? Okay, that's a little bit the question, and yeah, we're just gonna give it a try, I would say. I'm looking for my pipette again. Okay, and what time is it? Yeah, it's already 1 hour and 20 that I'm here. Yeah. You see, they're not able to move around a lot anyway because, let me see if there's a second one. They're not able to move around a lot anyway because I did not include a lot of water. Where's the other one? Here's another one. This one is, yeah. Actually also looks kind of nice. By the way, if you've never seen those guys before, these are water crustaceans, and the heart is here on the back. Yeah? Yeah, and they're sucking in water and debris, and this is the digestive system. You see that some green stuff is in here. Occasionally, you can actually see them see food moving through it. Yeah? Maybe in one of the upcoming videos, I'm going to add a little bit of yeast to those, and then we can actually see how it's actually eating the yeast. Yeah? So, but because this is a methyl cellulose live stream, let me add a little bit of this stuff here again. And let's see if this has an impact on those. I'm going to now go down with the magnification simply so there is a larger distance between the objective, otherwise it's too difficult for me to reach. I'm going to drop this on top here. I'm going to find it again. Where is it? And maybe this kind of slowed it down as well. More light. Oh yeah, actually it did. That's quite nice. Okay. Well, let me try this again here. Add a little more maybe. I've also not tried this out before. I hope I didn't. I hope I did not accidentally pick it up now with my pipette. Oops, I think I might have accidentally picked it up with my pipette. Oh. Okay. So this is basically what happens. But maybe this, here's the other one. Oh yeah. A little bit. Rotifer. How many of you can see the rotifer? Yeah. The rotifer is the one on the right side, the little guy. Yeah. The rotifers are also micro animals. Yeah. They don't move around fast anyway, so yeah. There's no specific need to slow them down. Let me see if I'm able to find it again. Here we go. Yeah, I think this one didn't really have a lot of the... Let me try this again. This is the last try. If it works, it works. If it doesn't work, it doesn't work. drop this directly on top here. Ah, that's the problem. It's so viscous that here we go. Oh, look at the heart. It's very beating, but the rest is, yeah. See, it's not even able to move its antenna properly anymore. Yeah. Okay. I've been able to observe Vorticella in a hay infusion several times. Ah, yeah, yes. Hay infusion is if you take a little bit of dry grass, add a little bit of water. Yeah. Just be careful. Again, you have you're adding you're growing again a lot of bacteria as well this way. Huh? Just make sure that you add then a lot of alcohol at the end to disinfect everything. Yeah. So yeah, so we see that indeed. Oh look. Now it's really struggling because it's trying to flip its antenna up. It can't do that because of the high viscosity, but the heartbeat does not seem to be very affected. Yeah? Did you manage to get your name on a new bacterium like Staphylococcus microbehunter? The thing is, okay, I'll talk a little bit about the naming of bacteria. The thing is, in the field of microbiology, you are allowed, if you discover a new species, you are allowed to name it, to give it a name, but you're not allowed to name it according to yourself. Okay, this is considered bad style. You can name it in honor of somebody else, but not you're not allowed to name them according to yourself. That is a little bit it's a tradition in when naming organisms. Yeah. So okay, let me see if there are some remaining questions. Otherwise, I'm just going to call it quits for the day. Yeah. As a little summary for those of you who joined in late, we started out by putting some paramecia under the microscope, and I added this methyl cellulose to it, which seems to work quite well to slow down organisms because it's very thick and viscous without actually harming them. So because osmotically not very active, so. We tried it with paramecia, tried it also with those nematode worms, and now last but not least also with those Daphnia, which are water fleas. Yeah, which generally I think it's okay if you if you just use a little bit of water, then they are not able to run away anyway. But we can see that actually it also has an effect on them. And I think what I'm going to do now is I'm just going to save it again, and I'm just going to put it back into the into the jar. Yeah, I mean, there are hundreds of them in there, but you see, a little bit out of principle and just because we're nice people, biologists, wants to, of course, also honor the organisms that we're observing. So what I'm going to do now is I'm going to take a little bit of, I'm going to go up here with the volume. I'm going to take up a little bit here, and I'm going to simply dilute it so that I can easily pick it up again. Yep, it's I think it's gone. Is it gone? And we put it back. Let's try it here again. Yep, that it seems to be. And let's try the last time. Okay, I'm quite, so, and we saved it again. And I'll be adding again, I just want to show you, maybe just as a last little thing, just want to show you how I'm feeding them. I've got this, maybe I can show you. I bought myself these tablets. These are food additives, which are chlorella algae, and dried. I don't know why people would actually eat those, but my little animals like it. So what I'm doing, I'm usually scratching off some of this, and you can see maybe that there's this powder falling in here. Yeah. And this is now the food. That's more than enough. And that's now the food for those water fleas. And occasionally, what I also do is I will add a little bit of, where is this? Ah, here it is. I'll add a little bit of the yeast as well. Just a little dry yeast, and now they're going to be happy for the next couple of days, and that's how I kept this jar alive for several months now. So maybe once a week or so I'm adding a little bit of this food, and I've seen that they actually start reproducing quite a bit. And then every now and then, when the water evaporates, look what I'm doing. I'm adding a little bit more water as well. And this is how I kept them alive now for, I don't know, maybe already almost a year. So they're always a good source. For things to put under the microscope. So let's see, last thing. Let's see what we've got here again. Yeah. Otherwise, I'm going to call it quits for today. Let me see, just checking again. Ah, yeah. This was the... Yeah. Where are they? I was just checking some of my old slides now. Last thing, just because I'm a little bit curious of whether they dried out completely. Yeah, they're... Ah, here, that's one of those Paramecia again here. Okay, still around. Let's see if the cilia are still moving. Or if it's... that last try. Yep, the cilia are still moving. It is still alive. Okay, yeah, I think is 90% pure ethylene alcohol good to clean my objectives? Is it the best they will sell me? Basically, pure alcohol can be used, but the thing is why do you actually want to clean your objectives? Unless there's a good reason, you should not be cleaning your objectives, in my view. And there is a certain technique on how you can clean. Just make sure that you do not put any of the alcohol directly on the objectives. You always have to put it on the lens paper. Yeah, it's really important that you do not have any alcohol running into the objective. Yeah. Okay. I think enough for today. Okay. Where is it now? Where is it? Where is it? Where is it? Oh, here's another one. I don't know. It kind of swam away. Here it is. Here it is. Okay, here we go. Yeah. So enough for today. Hope that you liked it. I wish you all the best. See you again next week. But in 2 weeks, I might have to change the date because I think in 2 weeks on a Saturday, I don't have time. So I might have to move the live stream. But I'll keep you informed. Please check the community posts. I'll keep you informed on any possible changes in the scheduling. Okay. But generally, I want to keep it at the same time. But I might have to make an exception in two weeks. Yeah. Enough for today. Wish you all the best. Happy microbe hunting as always. See you around next time, and bye bye. All the best.